Good morning, all of you. Most respected Professor Govindji, Professor Denny Thomas sir, respected Professor Jos Putul, Dr. Shivkumar, and other respected teachers, research scholars, dear students. Now we are moving to the second session of the last day of international webinar entitled Plant Functional Biology under the auspices of Doctrina. Today we have a Professor Dennis Thomas, the eminent professor. It's my privilege to introduce Professor Dennis Thomas for delivering an invited talk on the topic triploid plants from endosperm, an overview. Dennis Thomas, currently working as a professor in the Department of Plant Science, Central University of Kerala. He is an eminent professor having the specialization of plant tissue culture and somatic cell hybridization techniques. He did his PhD under two famous supervisors, Professor Bojwani and Professor Bhatnagar from University of Delhi, on the topic in vitro production of haploid and triploids of mulberry. He did 3 postdoctoral degrees in his academic and research career. First from University of Hamburg, Germany, on the topic in vitro fertilization with isolated gametes. Second from Hokkaido University, Sapporo, Japan, on the topic Alströmia tissue culture and protoplast regeneration. Third postdoctoral from Medical University, Rolako, Poland, on the topic secondary metabolite production in Augustine. His international recognitions are Boyska's Fellowship 2006-7 to carry out advanced research for period of 1 year at Department of Botany, University of Hamburg, Germany. INSA Visiting Scientist Award 2006-7 to carry out research in Biological Research Institute, Zebag, Hungary. INSA Visiting Scientist Award 2008-9 to carry out research in Field Science Center for Northern Biosphere, Hokkaido University, Ita, Japan. INSA Visiting Scientist Award 2013 to carry out research in Department of Pharmacological Biology, Medical University of Warsaw, Poland. Also, he is a visiting professor of Hokkaido University, Japan, to carry out advanced research and teaching in Field Science Center for Northern Biosphere. He has completed several national and international research projects with the support of different funding agencies like DST, UGC, CDB, and KSHEC. He has published 95 research articles in international and national peer-reviewed journals with a high impact factor. Moreover, he has delivered lectures in several national and international conferences. He plant biochemistry and biotechnology, Indian Mycological Society, and life member, International Society for Plant Morphogenesis. He has produced 5 PhDs and adjudicated several PhD theses. At present, 7 research scholars are working under his guidance. He is one of the editor of Botanica magazine of Delhi University, Botanical Society for a period of 3 years, and also associate editor of International Journal of AOB Plants, published by Oxford University Press. By all respect, on behalf of Department of Botany, Syed College, and the international webinar series Doctrina, moreover, on behalf of participants of this webinar, I proudly invite Professor Dennis Thomas for delivering the invited talk on the topic triploid plants from endosperm, an overview. Welcome you, sir.
Thank you. Thank you for your kind words. Thank you, Govindji. Thank you, Jos. I start my talk. The topic of my talk is triploid plants from endosperm, an overview. This is an entirely different area on which I did my PhD in University of Delhi.
Sir. Yes. Sir, can you put on the full screen, slide share? Full screen?
Yeah, sure.
Okay.
Is it okay?
No. Yeah, it's okay, sir. Okay, please.
Fine.
Okay, fine.
So I can start, I think. Okay, let us start with the basic. What is an endosperm? It's basically a nutritive tissue, usually present in the seed. And the endosperm in angiosperm is basically, therefore, if you can exploit this, if you can exploit this triploid tissue, you will have immense potential for developing triploid plants in nature. So usually, triploid plants are not much exploited or not much studied. Therefore, I choose this area for my talk. So, endosperm. It's usually formed as a result of what we call the triple fusion, the fusion between three haploid nuclei, one from the maternal side, two from the maternal side, and one from the paternal side. So this process is usually referred to as a triple fusion, the fusion between three different haploid nuclei, and therefore the product formed as a result of fusion between three haploid nuclei form the triploid tissue, that is known as the endosperm. So basically, it's a nutritive tissue. It's the endosperm represents about 60% of Earth's food supply. In cereals, the endosperm consists of mostly storage proteins, which form a staple food. And if there is something happens during the formation of endosperm in seed, so naturally the development of the embryo will be stopped, and thereby ultimately the abortion of the embryo may happen. Coming to the various types of endosperm, we can divide the endosperm into two different types. The exalbuminous endosperm, where the endosperm is totally consumed during its development, and in a mature seed, you won't get any endosperm. Only you can see the embryo only. In such a case, you can see in cucurbitaceae. So such seeds are usually referred to as non-endosperm seed. Whereas in the second case, albuminous endosperm, persistent. So in a seed, in a mature seed, if you dissect that seed, you can see plenty of endosperm. Rice is an example, where if you dissect a mature seed, you can more than 90% of the seed consists of endosperm. And therefore, such seeds are called by the name endospermous seed. Whereas in some families like Orchidaceae, Podostimulaceae, and Trapeaceae, mostly these members, the members of this family, doesn't have endosperm in its seed. So these three families are important in that respect. Coming to the use of triploid plants, we are talking about the production of triploid plants through endosperm culture. So before going into details, let us see what are the uses of these triploid plants. So usually when you compare the size or biomass of those plants which are triploid, they are having a more greater biomass, and also more stress resistant, and also larger organ size when comparing with that of diploid counterparts. And they are more economically valuable, and it's also used for developing new varieties. And for example, in case of phlox, which is an ornamental plant, the triploid plants produced were found to be having more thicker stem and thicker and larger leaves, bigger flowers, and bigger pollen grains, and even stomata are comparatively larger when comparing with triploid plants are usually seed sterile. It usually don't produce any seed. So such a quality is important in case of fruits like banana, grapes, apple, etc. So such fruits which is having no seeds are considered as more elite foods. Therefore, the price of such foods are higher. So another property is triploid plants are having more vigorous vegetative growth than that of diploid counterparts. For example, in triploid populus, this plant is usually used in the paper industry for producing paper. So the pulp quality of triploid poplar plants are considered as superior over their diploid counterparts. And also the triploid tomato produced were found to be more larger and tastier fruits, when comparing with that of natural triploids. Coming to how usually it is produced, the triploid plants are produced through conventional methods. Before going into tissue culture procedure, we have to just see how it is produced through conventional crossing procedure. Usually, triploid plants are produced by crossing a diploid and a tetraploid plant. So the first step towards this experiment is to produce a tetraploid plant. Usually, tetraploids are produced by treating the germinating seeds or vegetative buds with a chemical known as colchicine, which is usually used for duplicating the chromosome. So a diploid plant, from the diploid seedling or vegetative buds, you will get a tetraploid plant. But this experimental success in this experiment will be very low, just like 70-22% in various systems. And also it is lengthy and laborious. And once you get the tetraploid plants, then next step is to cross tetraploid plant with a diploid plant. So in such cases, the seed set and seed germination is very important. But unfortunately, in successful cases, the seed set and seed germination are very low. And the survival rate of such seedlings are also low. Therefore, and also it may show segregation also, since it is produced as a result of cross-pollination. So therefore, these are some of the difficulties when you go for a conventional crossing procedure. And in case of diploid plants produced through endosperm, such difficulties can be overcome. And it is considered as a direct one-step procedure for the production of diploid plants. So you can avoid all these crossing procedures and time by using endosperm to produce. So that is the advantage of using this technique. And also, as I already mentioned, the seeds are the diploid plants are seed sterile. Therefore, you won't get the seed. So in tissue culture, you can multiply these diploid plants by micropropagation also. Thereby, you can increase its number. So, regarding the endosperm, the history of endosperm culture, the first in culture was attempted by Lambe and Mills in 1936 in Siamese. They used potato extract and corn extract for using endosperm culture. And although the experiment was not very successful, they got minor callus proliferation or minor proliferation of the endosperm cells. Later in 1949, Larry, for the first time, reported the possibility of obtaining continuously growing callus from cultured maize endosperm. So during the initial stages of the development of this area, usually maize was used as a model system for developing the callus endosperm. So later in 1965, the direct plant regeneration from endosperm. That was the first report by Jory and Pojavani in 1965 from Department of Botany, University of Delhi. They worked with the system Exocarpus pupaciformis, a member from the family Sandalaceae, to obtain a complete plant from cultured endosperm cells. So at present, we have almost 80 various genera where the endosperm culture has been successfully reported. Complete plantlets were regenerated from various genera, about 80 genera. What are the factors controlling or affecting this endosperm culture? First thing is at what stage you have to culture the endosperm. So in this case, we have to measure the days in such a manner that it should be like days after pollination. So in various systems, we have analyzed after how many days of pollination we have to collect the endosperm from the seed. Days after pollination is very important for getting the better response. So in case of mulberry, it's 7 to 10 days after pollination. Whereas in Siamese, 8 to 11 days after pollination. And in Hordeum vulgare, 8 days after pollination. So this, all these cases, days after pollination, collection of endosperm is very important. Otherwise, we may not get a good response. And when you culture the endosperm, there are two types of endosperm, like mature endosperm and immature endosperm. In some systems, mature endosperm always respond when comparing it to that of immature endosperm. Examples in Lycopodium, citrus, malotus, and asparagus, a mature endosperm only respond. Immature endosperm, if you culture it, may not respond, and therefore, in such a systems, mature endosperm is preferred over immature. So this is a picture showing the endosperm-derived callus organogenesis in Imblica officinalis. So here you can see the picture with the endosperm callus showing the shoots emerging from such a endosperm. And in other systems, like Directa, Morus, Siamese, Englands, etc., immature endosperm only respond. So if you culture the mature endosperm, in that case, it may not respond. Therefore, it is a highly sensitive when you go for endosperm culture, whether the mature endosperm or immature endosperm, that you have to standardize in a system for getting good response. And in systems like actinidia, immature or irrespective of immature or mature endosperm, it responds. And usually, if you culture endosperm in a very early stage, it will not respond because endosperm is in a free nuclear stage. Therefore, it's often preferred that endosperm after 5 days or 8 days after, then only it will form a cellular type. Then only it can respond. Otherwise, these cells are in a nuclear stage. So this is a picture showing the endosperm at an early stage in this picture and a late stage, a mature seed. So the difference in these both pictures is that in an immature seed, majority of the seed, the endosperm is the major part. Whereas in a mature seed, since the embryo enlarges, embryo grows, the amount of reduced. So in a mature seed, majority of the endosperm is consumed by the developing embryo, and thereby it increases in size. As a result of that, the amount of endosperm reduces. So this is usually happens, and that in a non-endospermous seed, the endosperm is fully exhausted by the developing embryo. Therefore, in a non-endospermous seed, there won't be any endosperm. But in this case, in the second picture, it is an endospermous seed. Therefore, endosperm as well as embryo is present. And while going for culture, some extra additives like yeast extract, casein hydrolysate, and tomato juice, etc., used in addition to plant growth regulators for getting a good response. So in various systems, these extra additives were tried and found successful. This is a picture showing the endosperm regeneration in mulberry. the endosperms were cultured and callus obtained from such endosperm were substituted for shoot organogenesis, and such shoots were rooted and transplanted to soil. And later, it is the triploid nature of such plants were confirmed by cytology and also by stomatal analysis. So medium is also an important part when for getting good result. So mostly MS medium is used for culturing the endosperm. And during the initial stages of endosperm culture, that means the early reports, White's medium as well as MG medium, Urashik and Tucker medium also used. But nowadays, invariably, MS medium is used for culture. So, and in majority of the cases, for getting callus from endosperm, we use 2,4-dichlorophenoxyacetylcholine, callus induction from culture. And in some cases, auxin cytopenic combination is also used for getting callus induction in plants like parasitic, especially in parasitic plants like Taxillus and Dept. Auxin cytopenic combination is callus induction. And time varies from 10 to 20 days for initiation of callus from the endosperm. So this is a picture showing the endosperm organogenesis from rice explant, rice endosperm. So here you can see the callus developed from endosperm, and that calli produces the rice plants, and the acclimatization and transfer to soil, such triploid plants, and the triploid nature of such plants are confirmed by cytological analysis. In addition to these, there are some other factors like light, which controls the proliferation of endosperm. In residence, light is an important part, and without light, it will not develop. So normally we use 16-hour light period for getting response, but in some cases we have to give more light for getting proliferation of the callus. But in maize, it was found that light is not promotive, and dark condition is favorable for getting better response in maize. And another important factor for callus proliferation is the embryo factor. So in this case, we have to culture endosperm with embryo as well as without embryo to find out what is the difference. So in such cases, the association of embryo with the endosperm is only for a short period, like just maximum for 1 week or less than 1 week. So when we culture the endosperm with embryo, usually the embryo germinates and it will become green in color after 4 days, 6 days, or even after 7 days. So during this period, what usually happens is some useful plant growth regulator type substances will be coming out from endosperm, and that will help the endosperm further proliferate. So the association is only for less than 1 week, but it still was found to be helpful in most of the cases. So an early association of endosperm with embryo was found to be more helpful in some systems. For example, just like neem, usually this association of endosperm with embryo is useful in immature endosperm culture, whereas in neem, in mature endosperm culture, whereas in neem, even immature endosperm culture, it was found useful. Or usually embryo releases certain useful substances, gibberellin-like substances, which will promote endosperm proliferation. So this embryo fact can be overcome by using the GA3 itself. In the systems like Croton and Putunjiva, adding GA3 in medium or pre-soaking the endosperm in GA3 can avoid this embryo factor. So here a picture showing with embryo. So this is picture just show you a stained picture with the safranin endosperm. This is endosperm of mulberry and the embryo. So the same cultured in medium shown in the second picture. After 6 days, see here the embryo germinated and turned green. So at this stage, usually the embryo will be discarded and allow the endosperm to germinate or proliferate further. In this picture shows the origin of Asa directa indica endosperm derived plants. So this is the endosperm callus and the shoots originating from endosperm, and a single endosperm derived triploid plant, rooted plant shown in this picture. And the cytological analysis of such a plant shows the triploid number of chromosomes, and these are the acclimatized plants showing well-developed plants here. And these two are the pictures showing in some plants. It shows certain chromosome variations, not exactly a triploid number. Some variations are also plants. So we were talking about the endosperm callus. Now, once when you get the callus, you have to go for organogenesis. That is for getting shoots from the callus, endosperm derived callus. But in case of the first report in 1965 by Yori and Botany in exocarpus, there was no callus formation. Only thing what happened when they cultured the endosperm was the direct shoot induction from such a callus, such an endosperm. So there was no callus formation. The endosperm directly gave rise to shoots. So direct shoot induction from callus was direct shoot induction from endosperm was obtained in the first report by Yori and Bojavani. And they obtained shoot buds all over the endosperm surface. And the medium was white medium and supplemented with IAA kinetic casein hydrolysate. The omission of casein hydrolysate in the medium actually increased the shoot number to 13 to 26. So here's a picture showing the endosperm regeneration in Carthamus. So here you can see the endosperm derived callus, and here the shoot organogenesis from such endosperm derived callus and individual shoots you can see. So when you observe pathway of shoot induction from endosperm, it may be a direct shoot induction, as the first report. Just when you culture the endosperm, the shoots are emerged from directly from the cultured endosperm. So that is known as the direct shoot induction or shoot bud differentiation. Examples in Exocarpus, Taxillus, Leptomeria, Screwula, Dendroptera, etc. Direct shoot bud differentiation. Whereas in some other systems like Morus, Jatropha, Putunjiva, Podium, and Malus, shoot buds from endosperm callus was reported. So in this case, in the second case, the cultured endosperm proliferates and produces callus. And that callus, when you subculture on a suitable medium with plant growth regulators, produces the shoots. So in addition to these two, this is a picture showing Asa directa endosperm organogenesis, callus organogenesis. So this is a picture showing the seed. Here you can see the embryo in green color, and these are the endosperm, and this is the seed pod. So this endosperm cultured with embryo. This is the, you can see here with the green color embryo here, and this embryo will be removed after 1 week, and then the endosperm derived calli proliferates and produces several shoots. And here you can see the shoot buds emerging from such a endosperm cavity. Another picture showing in Sapium, the endosperm derived shoots. Here you can see the shoots. These are the endosperm. This is the embryo. So such endosperm produces the shoots, and these are the triploid shoots. This is a rooted shoot, and subsequently they were transferred to the field for activities. So this is another picture showing the mulberry endosperm derived plant. And in that case, when you compare the shoots, this is a shoot, endosperm derived shoot. When you compare this shoot with normal diploid shoot, these shoots are comparatively larger in size, and they are more thicker also. So by visual observation also we can see the difference between the diploid and the triploid plant. So we were talking about the various types of shoot organogenesis. One direct organogenesis, another through callousing. And in rice, immature endosperm underwent two different modes of differentiation. So that means one system can even respond differently in various media. For example, in rice, direct regeneration from the explant, that is from the endosperm, and also indirectly via callus formation. So in the same system, you have two different pathways. And in addition to this indirect and direct pathway, there is another pathway known as through endosperm embryos. So in that case, what usually happens is when you culture the endosperm, it proliferates and produces callus initially, and such calli on subculture produces embryos. So that embryo is usually referred to as endosperm embryos to differentiate it with the normal somatic embryos. So endosperm embryos were reported in systems like Nigella, Petroselene, Citrus, Prunus, Santalum, and Juglans. So this is the third pathway of endosperm development, endosperm derived plant development. So this picture shows the endosperm embryos in Juglans. You can see here the embryos. And this is a picture showing the cytological analysis confirms triploid nature. Now coming to what are the various growth regulators. Hormone usually supports callousing as well as shoot organogenesis in various systems. In mulberry, MS media supplemented with BA and NAA, and also casein hydrolysate and yeast extract often supports callousing. Whereas for shoot induction, BA and NAA or TD alone produces or give rise to shoots. In neem, for callousing, NAA, BA, and casein hydrolysate is used. And for shoot induction in neem, BA and kinetin often helpful. And in actinidia, for callousing, 2,4-D and kinetin give better callousing. And for shoot induction, MS medium supplemented with the trialdestrone was found. This is another picture showing the shoots of endosperm derived endospermous plant. And here, the histological analysis of the callus while developing the shoot. Here you can see the shoot buds originating deep inside the callus mass. So this is an early stage of the shoot bud, and little bit mature shoot buds here you can observe. So by observing histological analysis, we can find out whether the shoots are coming from the base or from the boundary of the callus or from deep inside. In Acacia nenotica, for callousing 2,4-D, BA and casein hydrolysate found helpful along with MS medium. And for somatic embryogenesis, in this system Acacia, we obtain somatic embryogenesis. And here there is no change in the medium as well as the plant growth regulator. The same medium as well as plant growth regulators were used for getting somatic embryo or endosperm embryo. So there is no change in the medium. Only the subculture is continuously, and during the third subculture, it produces the soil. So there is no change in medium. Only the same medium was, and the explants were repeatedly subcultured. So initially, it produces the callus, and callus on subculture on the same medium. During the third subculture, it produces. And in petroselenium, callusing and hemorrhagenesis is observed in a basal medium. It did not need any plant growth regulators. Only basal medium was sufficient for getting callusing as well as hemorrhagenesis. This is the picture showing endosperm embryogenesis in acacia. So this is the cultured endosperm. Here it produces the callus on the surface of the endosperm. And this callus, after subculture, produces the endosperm embryos. You can see here many embryos in this picture. And such embryos were after subculture and germinated and produced the pupa. And in citrus, another system, hemorrhagic and Tucker medium or White's medium was found helpful or found useful for obtaining a callus along with 2,4-D and cation hydrolysis. And in this case, the pathway was through endosperm embryos, and endosperm embryos were obtained in 2-mg medium. 2-mg medium means a medium containing double the concentration of normal mg medium. The 2-mg medium supplemented with GO3 produces embryos, endosperm embryos in citrus. In Malotus, 2,4-D and kinetic was found helpful along with MS medium. For callusing, for getting shoots, MS medium supplemented with BA and NA was found. So this picture shows endosperm embryos in citrus. The early stage of embryo development in citrus, this fully mature embryo in this picture. And embryo-derived plants you can see in this picture, and the triploid number of chromosomes after cytosis. Now, this is another picture showing the endosperm derived plants in papaya. So this is the picture showing the endosperm derived calli here, and the shoots derived from endosperm calli, and they were rooted and transplanted to soil successfully. So this is a recent report by a Chinese group. So they obtained triploid plants from papaya. You know the importance of papaya, where usually when you cut the papaya fruits, plenty of seeds are present inside. So usually such a triploid papaya, you won't see any seeds. So it may be a seedless fruit when you get papaya fruits from triploid plants. So usually for confirmation of triploid nature, we go for chromosome analysis. So we have to see what is the number of chromosomes, and if it is what is the number of diploid number. Then accordingly we will see what is the number of chromosome in the expected number. So if it is a triploid number of chromosome, we can confirm that it is a triploid plant. But additionally, there are some other experiments like stomatal analysis. So the triploid plants, the size of the gut cells will be larger when comparing with that of a normal diploid plant. So this is a picture showing the stomatal gut cell analysis. This is the diploid plant, and this is a triploid plant. So the photographs are in the same magnification. So the size of the triploid gut cells are comparatively larger in size, and also the chloroplasts present in gut cells are also large in number. So this is an additional proof for confirming the triploid nature of the endosperm derived plants. And in that case, the stomata number per area, per a particular area will be less in case of triploid plants and more in triploid plants. This is just an additional proof for confirming its triploid nature. So what are the problems associated with this system or this endosperm culture? We can culture either immature endosperm or mature endosperm. So if you go for immature endosperm, in some systems, nuclear endosperm is present during the early stages. So such a endosperm we cannot culture because it is in liquid form. So therefore we have to go for mature endosperm only in some systems, or wait for the endosperm to develop into a cellular endosperm. Then only we can go for culture. And if you get the get the endosperm, when you go culture it, it do not produce callus in some cases. So that is another challenge. So you have to standardize the medium as well as the plant growth regulators for induction of calli from such endosperm. So that is another difficulty when you go for endosperm culture. And usually what we do for endosperm culture, we have to isolate the seed from the field at the early stages. Then when you go for dissection, you have to dissect out the endosperm from the seed. Then in that case, usually some systems, the seed root is very hard. So dissection is a difficult process. So we have to do some softening mechanisms so that the seeds we can easily dissect out. So if it is a hard seed coat, that's another challenging factor for dissecting the seed to isolate the endosperm. And in cases where you get the callus, when you go for organogenesis or shoot induction from callus, in some systems shoots may not develop. So it's somewhat a recalcitrant system where you may not get shoots from the callus. So you have to standardize the proper medium, proper plant growth regulators. So even after standardizing all these, in some systems you may not get shoots callus. So that's another most important challenging factor when you go for endosperm culture. And another problem is regenerated plants from either from callus or direct regeneration. Mostly this shows certain abnormalities like fasciation. The shoots are like a flat. Usually the stems are round, but here it is shows certain abnormalities, and such shoots the growth is also standard growth, shows standard growth. So this type of abnormalities also seen in some systems, so that has to be rectified by standardizing. So another challenging factor is the chromosome variations. Normally, the triploid number you will get only very few plants. Other plants, chromosome variation like one chromosome, 2n minus 1, 3n minus 1, or plus 1, like that. Some chromosomal variations we also observe in some systems. So this is also considered as a difficulty in assessing whether it's triploid. And last slide regarding the prospectus. This procedure should be, as I have already mentioned, about 80 systems, 80 plant genera it is reported. So we have to focus on more systems. Weighing its difficulty, I think most of the people are not working in this area because you are facing a lot of problems. And therefore, you have to focus on this procedure or this protocol, then only we can get more results from the system. And the seedless in this we can focus this endosperm culture on more systems where seedlessness is used to improve the food quality. So in such seeds we can concentrate, we will get more good varieties we can develop more good varieties and economically useful plants by using this technique. So with these words, I conclude. I thank you very much for giving me an opportunity to share my ideas and knowledge with all of you. Thank you. Thank you very much.
Hello, sir. Thank you, sir.
Thank you.
Thank you, sir, for your wonderful presentation on the topic. Now it's a time for discussion. Govindji,
I think he put mute.
Yeah.
Okay.
Yeah.
Well, I'm here. It's a very exciting thing for me because I don't know anything about it, except I know Jory, B.M. Jory, I think you mentioned, and I also know Satish Maheshwari, Sipragua Mukherjee, and all those people who worked on it. And we just published a tribute to Satish Maheshwari. So
I saw it yesterday.
Yeah.
In Physiology and Molecular Biology of Plants, in that journal.
Wonderful.
I saw it.
Oh, great. So what I'm saying is my knowledge is as enjoying this work, but not able to technically be in it, but it's beautiful. That's all I would say. Thank you.
Thank you, sir. I think this session is very much useful for students as well as teachers.
Yeah.
Very nice presentation, sir. Sir, we have some questions, sir. Can we discuss?
Yeah, definitely.
Sir, first question is, what type of media is usually recognized for endosperm culture?
Media means usually nowadays when you examine the reports, the published reports, nowadays only MS medium is invariably used, along with various plant group varieties. But during the initial stages of this report, it starts from 1965 onwards. During these earlier reports, they used the Weiss medium, MG medium, etc. But nowadays it is a MS medium because MS medium is considered as a general purpose medium. So we can use it for any purpose. So usually in most of the reports, MS medium is invariably used during recent reports. But the earlier stages, other media like Weiss medium as well as MG medium, Morashik and Tucker medium also used.
Okay, sir. Sir, my next question is there any variation in gene expression in endosperm cell in vitro?
Actually, that type of study has not been reported so far, whether any change. But physical variation usually seen when you observe with the diploid plants as well as triploid plants. As I have already mentioned, it's large in size, leaves are more thicker, stems are also large, biomass is more, and fruits are also different. So that type of variations only so far studied. Molecular studies, I haven't seen any reports, but that has to be reported, I think.
Okay.
Variation.
Okay, sir. Thank you. Next question is, sir, what is the advantage of yeast extract while using in endosperm culture?
Usually, these all extra additives like yeast extract, tomato juice, casein hydrolyzed, all these extra additives are in most of the reports used for getting callus proliferation from the endosperm in the initial stages of culture. So for these extra additives were also used for getting shoots in some systems. But other systems usually for callus induction only they are used. For shoot induction they are not used. So mostly we can conclude that these extra additives like yeast extract, casein hydrolyzed, tomato juice, etc. are used for getting callus proliferation mostly. But some other systems is used for shoot induction.
Okay, sir. Sir, one more question. Sir, any morphological difference in shoot and fruit for this type of plants?
Yeah, definitely. Morphological difference means the size is higher, means more bigger size. Suppose we have a diploid plant and a triploid plant of the same age. So when you compare the size, the triploid plants will be having more size, physical size than that of diploid plants. One thing. We can put it in another way. saying that the biomass is more in well, I will tell you in another example so that you will understand. The morus, that is in mulberry, the use of mulberry is to feed silkworm for getting during the production of silk. So when you use a diploid plant, you can feed a less number of silkworm because its leaf size is less when comparing with that of diploid plant. So, but at the same time, if you grow the triploid plants in that same area, for example, 1 acre, you can feed more silkworm from that plants because its leaf size are more. Means size is large. Therefore, we can feed more silkworm. So that way, the biomass is more in triploid plant. What was the other part of this question?
Only that much, sir.
Okay.
Okay. So only this much questions we had in the chat box. I think if anybody want to interact, please. Otherwise, we can move on to our next session. Sir,
Okay, thank you very much.
Sir, on behalf of Doctrina 2020 and Department of Botany, I extend my our sincere thanks to you, sir. Thank you very much. So now we are moving to our